Thanks very much to One Nucleus for the invitation to speak today, and thanks very much also for the title of the talk. I'm not sure I completely understood it, so hopefully I'll be talking about something that's vaguely matches the title. But as I've been introduced, I work for MedImmune, which is just based in Abington, but it's in effect the old Cambridge antibody technology. So it's a sort of a nice follow-on from the from the previous talk. So what I want to talk to you today, there will be a little bit of science and a couple of case studies, is the concept of screening now with the focus in mind of making a product that's going to be manufactured and given to patients in the clinic and then commercially. And this has been termed in the biotech industry as developability. And I'm going to come at this from very much an antibody engineer's point of view. I'm an antibody engineer very much in the early stages of research. But the thinking behind it being that we have to work more closely with our development partners to really maximize the benefit that we can get in terms of being able to actually manufacture these amazing antibodies and other therapeutics that I guess we're going to hear about throughout the day. So I'll give you a bit of a context around that, and then a couple of examples with a little bit of data for two different ways that we currently are trying to assess and to screen for those molecules that have the appropriate characteristics in terms of biophysical properties for development. So I'll just start with I guess the basics. So it's pretty straightforward in principle to make an antibody or other biological therapeutic. You isolate your product, you make your product, you find the right patient, you put them together, you see the right effect in the clinic, you go into full development. So that's pretty straightforward, and I guess everybody who works in industry in here can relate to that pretty much is always how it happens. However, first of all, you've got to find the right candidate molecule. So as well as finding the right patients, which we all invest a huge amount of time and a huge amount of effort and resource into doing, clearly getting the right candidate molecule is important. And the previous talk really introduced some of the core technologies that have allowed that to happen. So one of the technologies invented, obviously, here, and good to see John in the front of the audience, was phage display, which was as we've just heard, the backbone of Cambridge antibody technology. And we also use ribosome display, sort of a complementary type approach. But the same principle can be found with transgenic mice or with humanization, in that by having a huge repertoire of potential protein variants that you can use to tailor to fit a particular characteristic that you're after, as well as combining that with some sort of screen to isolate those molecules that have that desired characteristic, you can really tailor what your drug wants to do in the body. So the typical things that we would look for would be that it's got to have the right target specificity. Obviously, you want to be your antibody wants to be binding to the right epitope and to the right antigen. Ideally, you'd like species cross-reactivity to help power pharmacological and toxicological studies. Obviously, you want something to be as potent as possible, so you would screen for both in vitro and in vivo potency. And with antibodies, one of the clear ways that you can optimize them is to increase the affinity for the target. And it's this sort of this interplay between these different characteristics that's the real key to getting the right molecule that you want. And it's that combination of having powerful libraries of different variants with the ability to design your screening cascade such that you can find those target clones that really have all of those different characteristics that's really driven the therapeutic antibody and other therapeutic biologicals forward in the last 10 or 15 years. So this, I guess, should be this sort of Venn diagram should be pretty straightforward and pretty obvious to most of the audience here, that by looking for these different characteristics simultaneously, we find the right molecules that we can take forward. And certainly 5 years ago, these would have been the sort of considerations that I, as an antibody engineer, would have had forefront in my mind. Would be the key things to design into a selection and screening cascade. However, we now know, as more and more molecules go through the clinic and through manufacturing processes, that production of biopharmaceuticals is very stressful. There's the bioreactor process and the fermentation induces a huge amount of different stresses to the molecule, as does the purification through the different stages of things like anion and cation ion exchange, removal of viral particles by low pH, importantly concentration and diafiltration. But I would also add to that things like filling pre-filled syringes at really high concentrations for subcutaneous delivery, which is becoming more and more important, particularly in areas such as rheumatology. And also an aspect that certainly within research is probably not really thought about too much, but actually the shelf life of the molecule. If your antibody is actually going to go into patients and is going to be produced commercially, it's going to stand on the shelves or in a fridge somewhere, and potentially for a large amount of time. And having an understanding of what can go wrong or what can change in that molecule is really important. And we now know throughout the industry of a number of examples whereby molecules that basically behave aberrantly and that can then cause a lot of additional cost in terms of both resources, time and money in terms of putting these things right and mitigating some of these problems. So certainly within the industry in the last 5 years, the approach is now being taken, how can we mitigate some of these production risks earlier? So if we go back to this idea of trying to screen simultaneously for the different characteristics, we can think about some of the issues that have become apparent over the last 10 years or so as more and more molecules have moved through the clinic and have become commercial. So there are things like the actual solution properties, how soluble your antibody or the protein is, whether it becomes viscous at a high concentration, the actual physical stability in terms of the thermodynamic folding and unfolding of your antibody or other biologic, the potential for aggregation, which can be a big issue for antibodies and other proteins, and then the chemical stability side, particularly this idea of shelf life, the different amino acid side chains that make up proteins have different post-translational modifications that can become apparent either under stress or over time, things like deamidation of asparaginins, oxidation of methionines and tryptophan, etc. So if you think about all of those different properties that potentially can prove problematic once the antibody actually moves further into development, I guess the model now is how can we, as early researchers, mitigate these as early as possible. And we've been working on a number, as I'm sure others have, of a number of approaches in the same way that we screen early on in our research for a particular antibody for things like specificity, cross-reactivity, affinity, and potency. At the same time, if we think about, well, can we screen at that early stage when we're still dealing with thousands of molecules for things like thermal stability, aggregation propensity, and post-translational modification? And we've implemented a number of different techniques, which I've just shown a few examples of early into our discovery process. And I'll talk about two of them, the CIF approach and our in silico aggregation prediction screening approach in a little more detail as a sort of a model. So if you think about it, what we're doing is adding more and more screens into this early process. And that isn't always an easy marriage, as you can imagine. Even with huge libraries of variants as we go through the selection and screening cascade, the more questions that you ask of a particular model and of a particular molecule, and the more filters you add, the more things you remove by the very nature. So you have to sort of balance these things in terms of the functionality versus what we're calling the developability attributes. And sometimes these will be in sort of opposition to each other. We've had a number of examples, certainly internally in-house, where increasing the affinity beyond, say, the single-digit picomolar level for really high, for a really high affinity, high clinical efficacy molecule leads to simultaneous reduction in the stability of that molecule and an increase in aggregation. So these things have to be balanced, and a sort of a risk mitigation strategy has to be put in place. So in terms of, I've just sort of given you a couple of examples of some of the screens that we've put in place, and I just wanted to sort of add where to, the question of when to perform these developability screens. So typically, as I say, maybe 5 years ago, we would have moved through something that looks like the pipeline, if you like, at the top of this particular slide. And if you look at the numbers of molecules that the researcher is thinking about, at the beginning, if you're working with some sort of combinatorial library approach, you might have 10 to the 11 different molecules, which when you go through the different selection cascades, you might go down into hundreds and the tens. Going through lead optimization via potentially affinity maturation, etc. You again will start with thousands and then down to hundreds and tens. And as you move through the different questions that you're asking of your molecule in terms of affinity, potency, and cross-reactivity, we would generally get to a point where we have 2 or 3 molecules that do exactly what we want, which we would then move over into our development and our CMC colleagues, who would then go through the scaling up and moving into preclinical development and then into clinical development. And at that point, you've basically locked the sequence. You've chosen your antibodies of interest and you've moved them into preclinical development. But it's at that point that often these sorts of issues will be seen. So when you're going with looking at high concentrations, you're moving into more research cell lines. At this point, issues in terms of manufacturability can only become apparent here. And this is quite an expensive time to be actually spotting these issues, because you then have to go through quite complex strategies for mitigating these effects, or you have to go through some sort of a more complex formulation strategies in terms of the buffers or the excipients that you have to add to the final drug molecule. Whereas if you can actually introduce this sort of idea of developability screening while you're still dealing with thousands of molecules, that gives you a better chance, we feel, of getting those optimal developability characteristics as early as possible, so that you can reduce the risk of these issues becoming more apparent later on down the line. So hopefully the benefits of this will be that at the very least, you'll allow early identification of any potential management issues, which is in the CMC world, which is chemistry, manufacturing, control, is known as quality by design model. But ideally, and what we're certainly moving to at MedImmune, is that developability screening will allow these unfavorable attributes never to actually get all the way through to the clinic or the preclinical model, where we're actually scaling up into the grams and the kilos of material required, that we will have filtered them out while we're still dealing with the thousands of molecules at microgram or mig scale. So having introduced that sort of idea of developability screening, I wanted to just go into a bit more detail into 2 of the approaches that we currently take now for all of our projects that we're looking at in terms of therapeutic monoclonals and other protein therapeutics. So the first of which is actually an in silico screen, so actually not a physical biochemical screen at all, but an in silico screening approach that we've added for all projects now, that's called AgPredict. And this is based on an algorithm developed at MIT by Professor Bernard Trout. And it's basically an algorithm that can predict, or that tries to predict, spatial aggregation propensities. And this is run at an atomistic scale. So every atom of every amino acid in a given molecule is modeled, and then molecular dynamics are performed such that the actual movement of the molecule itself is modeled. And from that, a ranking is given to potential sites of aggregation that could be found on that molecule as it goes through its motion. That algorithm was first published a few years ago now by Trout's group, and subsequently has been licensed into the Accelerus Discovery Studio, which is one of the leading protein modeling software packages, and that's now currently being run on every antibody therapeutic project that we run at MedImmune. And this is a good case study that hopefully will illustrate why it's important to catch these things early. So we recently developed a potential product, an anti-NGF monoclonal antibody. This was a really high affinity molecule. It was single-digit picomolar. It was mouse cross-reactive. It was very specific to NGF itself. It was the most potent. It was beautifully potent in all of in vivo models in the rodent. So within research, we felt pretty pleased with ourselves with having developed this molecule. We handed it over to our colleagues in development who hated it from day one. It wouldn't behave in their hands at all. It exhibited reversible self-association as measured by size exclusion chromatography, very poor solubility, crashed out of solution if you looked at it wrongly, high viscosity, formed a gel, low purification yield, separated, phase separation if you tried making any of it. It basically was a horrible mess. In a researcher's hands at small scale, it looked okay. It wasn't the best looking antibody in the world in terms of expression, but we all thought it was fine. Handed it over to our colleagues and made enemies for life. So we used this as a model to try and see whether, going the wrong way around, whether we could actually go back and see if we could make amends by re-engineering in some stability into this molecule and removing some of these aberrant biophysical characteristics. So we ran the aggregation prediction, so this in silico model, just based on the model FV and then plugging in the actual sequence for this. And we ran it on the parent antibody, which I should note did behave itself. It's a lower affinity, lower potency version of this antibody, but it behaved nicely and was very much considered developable by our colleagues in development. And then we ran our super high affinity version of this. And the algorithm you can visualize by basically, if something is really red, then it's a patch of high aggregation propensity. And if it's really blue, then this thing is considered to be modeled to be something that would be very unlikely to aggregate. And the FV you perhaps can't see here. It's just the front and the back view. The top half is the front view. The bottom half is the back view. You can see at the bottom there are a few of the FV model, there are a few sort of red patches, and that would be the area that would be in contact with the constant region. But at the top of the molecule, you can see that within the CDR portion, there are differing levels of aggregation, potentially aggregation-prone sites in the molecule. And you can see, looking at slightly differently from a sort of a bird's eye angle, on the left again you have the parent molecule, which most of if you like, the aggregation propensity regions are in the actual CDR3 region. But actually, on the right, when we have our higher affinity variant, we've introduced a huge patch across multiple CDRs of potential aggregation propensity. I'll just check my timings. So we ran a number of different screens. Initially, we looked at things like thermal stability and whether these things, so we made it, so I should stop a second there. So we basically took that information, found which sites were potentially contributing, and made a series of mutants back to the parental version, and then tested them. So here on this slide, we just see some high throughput thermal stability data and simple aggregation propensity data that shows no difference between any of the molecules. So whatever was going on to to cause this antibody to be a problem antibody wasn't due to just thermal stability, to it unfolding early, so it was something else. What we then did was look at some size exclusion analysis to see whether it was aggregation or fragmentation. So this is quite a busy slide. So on the left, we basically have just a reference and a standard antibody that we would use for SEC just to show what a monomeric IgG profile should look like. Next down on the second level, we have the parental antibody, which again, although shows a little bit of aggregation, is still largely the monomeric peak. And on the bottom left, you can see the mess that our antibody of lead gives when you try and run it down an SEC column. And on the right and on the next slide, you can see that just 3 mutations that we've made that have been guided by the AgPredict algorithm have absolutely rescued this monomeric peak. So we can see that just by targeting these 3 residues, and we can actually go even further, that actually it's one particular tryptophan in one of the CDRs that if you revert back to the parental sequence, absolutely gives you a really nice monomeric peak on the SEC. So taking that together, we made a number of mutants and chose one of the mutants, STT, which basically just represents these 3 amino acids that we've changed back. This is pretty hot off the press data, but we've seen that certainly at a small scale that this antibody behaves beautifully. Subsequent to actually putting this slide together, we've actually got some higher concentration data, and actually it's a lot more soluble. There's no phase separation. The viscosity is back to normal for a standard antibody, and it basically behaves as one would hope for. There is a slight loss in potency, but it's within sort of 3 to 4-fold. So actually, it's still over 100-fold better than the parental starting point. And for the purposes that we require, has pretty much rescued the biophysical properties whilst maintaining just about the affinity and the potency of this molecule. So I think it shows the power that using this sort of in silico guided mutagenesis can really sort of tweak the developability characteristics even of a problem antibody. But I really wanted to just to show you, unfortunately I can't tell you what the antigen is in this case, but that was really the worst case scenario of taking a problem antibody or a problem biologic and trying to rescue it using this sort of in silico approach. The second sort of case study I can show you here, where we actually, live in a project during the research phase, we actually screened in silico the variants that we were making via lead optimization. And you can see here for this recent project, so on the top part of the slide is just 4 models that have been generated by the AgPredict of 4 different variants during an affinity maturation experiment, and we can see that there's a really nice correlation with both behavior in size exclusion chromatography and also in reversible self-association as measured by analytical ultracentrifugation, which is really the sort of the gold standard technique for looking at that sort of non-specific interaction of molecules, of the same molecules in solution. And we can see here a really nice correlation between the predicted behavior in silico and the actual physical behavior in the lab. So that's a technique that we're now, as I said, instigating onto all therapeutic projects, and that we're really happy with. I just wanted to quickly finish up with another example, and this is looking at a high throughput assessment of chemical degradation. So as I mentioned earlier, one of the issues that can become apparent as you take antibodies into the clinic and further on into development is this idea of shelf life and the potential post-translational modifications that can take place over time or under stressful conditions of particular amino acids. So we got together with a company called ProteinSimple who make a capillary isoelectric focusing equipment, but this is a high throughput version that can basically be run at 384 well. And we carried out an initial assessment, and we've subsequently got a couple of these machines in and have now implemented them. Because the nice thing about this CEIEF technology is that it can, because it measures changes in charge, for things like deamidation of asparaginase or potential end-link glycosylation that can occur with a particular antibody. This thing can spot it at really high sensitivity, so it just needs sort of femtograms of material. So initially we wanted to try this out because we thought it looked quite interesting, could potentially be used as a high throughput screen for chemical degradation of our molecules. And so we carried out a trial experiment with two antibodies, one of which we knew there was a deamidation that happened at the VHCDR2 in one of the asparagines, that when that gets deamidated to isoaspartate and then aspartate, we get a significant loss of potency and affinity. So clearly that residue is playing an important role in the biological effect and the binding to its target molecule. So we knew we had a nice test example of something that rapidly gets deamidated, and there's a functional consequence of this. And we also had an antibody that we knew behaved itself really nicely. So we took different samples from the different stages of the discovery and the development process, going from the purified IgGs at the end all the way back to the early data. And what I wanted to show you here is basically what an IEF trace generally looks like for an antibody. So you have a main peak, and then you have a basic variant and an acidic variant due to the PI and whatever buffer that you're in. And we could see that for MAB1, which is our antibody that you would basically shouldn't have any sort of deamidation going on. It's a nice stable antibody. You can see you get this large main peak and just a few minor peaks of the different charge variants. And at MAB2, which is the antibody that we knew was deamidated rapidly, you can see this more heterogeneous profile of the two antibodies. And this was with a real small-scale 96-well IgG analysis. So this is something that we can implement within our research screening cascade. We then looked to see whether this sort of profile, how it matched to if you like, the gold standard sorts of IEF analyses that are run later on in the development process, and we saw a really nice correlation between all of the different profiles that we saw. So if you like, the more heterogeneous antibody that we know is prone to this sort of chemical instability gives a really similar profile, as does the well-behaved antibody. So this convinced us that this was a really nice potential screen, which again we've now implemented across all therapeutic antibody projects, that as well as looking at all of those different physical stability in terms of screens that we can set in, we now look at the chemical modifications while we're still in the sort of the thousands of different antibodies stage. So I'll finish up there, and hopefully I've convinced you to a certain extent of the importance of screening for these biophysical properties as early as possible, and give you just a snapshot of a couple of examples with real data of where these are now being implemented in the actual therapeutic antibody generation projects in-house. So with that, I'd just like to thank all of the contributors and you for your attention. Thank you.